It is started.
Let me check. I will inform you.
Yes. Okay, so dear all, good evening to one and all. I am Mr. Durgesh Toliram Gautam, Assistant Professor and Training Placement Coordinator. I heartily welcome you all for this Alumni Talk Series 1. On the behalf of Yeshwantrao Bhosle College of Pharmacy, as well as the Training Placement Coordinator, I welcome all the participants for this Alumni Talk Series 1. I would like to welcome our Principal Honorable Dr. V.A. Jagtap sir. Welcome, sir. Again, I would like to welcome Dr. Dattatray Shinkar sir, HOD, Department of Pharmaceutics. I would like to welcome Mr. Tushar Rukari sir, Assistant Professor, Department of Pharmaceutics of our college. And again, I would like to welcome Mr. Vinod Mode sir, HOD, Department of Pharmacology. Again, I would like to welcome all the today's our speaker as well as the our eminent alumni for this today's program, that is Alumni Talk Series 1. Today with us Miss Pritam Palkar, Miss Prajakta Kashadikar, and Miss Mona Mohite. I heartily congratulate to all of you first, especially for qualifying the GPAT examination 2020. My best wishes for your upcoming future. Then I would like to welcome all the participants of our students as well as the students from other institute for this program, that is Alumni Talk Series 1. So training placement of our cell or our college, training placement cell of Yeshwantrao Bhosle College of Pharmacy organized this Alumni Talk Series 1 simply for giving us some guidance and some sharing some experiences regarding the how to prepare for GPAT as well as the NIPER examination. So definitely that experiences as well as the guidance definitely will be helpful for those who are the GPAT aspirants as well as the NIPER aspirants. So without taking much time, I would like to share, or I would like to request our eminent, we can say, alumni, Miss Prajakta Kashadikar. You can start your program. So over to you, Prajakta. Hello, Prajakta. Over to you. You can start. Yes, Prajakta, are you there? Hello, Pritam. Suppose I think yes, I think Prajakta, she is having or facing some internet connectivity or issues are there. Better you should start, Pritam. Hello, Pritam. Am I audible, Pritam? Hello.
Hello.
Pritam, you should
Pritam,
start.
you should start. Okay. So basically, my part about guiding you all for GPAT on IPER would be telling you all more about what topics to be covered or which books to be referred for your preparation for GPAT. So I'll go subject wise. If you all want to note it down, you all can. So first, my most favorite subject would be APP and pharmacology. So talking about APP, I would tell you all that prepare a chart about various pH like salivary pH or pH of your stomach, pH of intestine. So you all should prepare a chart regarding what are the different pH of body fluids. Also the composition of different body fluids, what kind of fluid it contains, carbohydrates, proteins, etc. Like for example, composition of bile juice. That is very important in case of APP. Next is also composition in terms of like uh, stomach, which sort of tissue stomach is made up of, or which muscle is present in which part of body. So this anatomy, certain parts of anatomy are important in terms of GPAT. Also bones in case of like how many what are the total number of bones present in your body, like what in vertebral column there are different parts. So even taking an example of thoracic vertebra, so what is the number, where it is situated, what is the position, those points are important in case of bones. Then in case of heart, anatomy is really very important along with its functioning. So like different names, like we have bicuspid wall and tricuspid wall, but bicuspid wall is also called as mitral wall. So such questions can be asked, and these basic things you all should know regarding APP. Next, I would say about gynecology and medicam, that I would tell you later. Next is pharmacognosy. So in pharmacognosy, I would suggest you all to refer the Kokare book which we use for practical. That I think greenish blue color book is there, Nirali Prakashan. I would suggest you all this book because it has properly given the main aspects of cognosy which are important and only which are essential in case of GPAT or NIPER. So because it has division like root, fruit, stem, and leaves. So when you all do a group study, you all can distribute within you all. Like today, I'll study about roots. So another person will study all the drugs which come under fruit, and another person will study about stem. So while studying this, you all have to make sure that you all jot down your own notes. What notes in kind of like everyone has a different pattern which I followed, or we three of them, like Akshata Thakur, Prajakta Kashaikan, and myself, we had done a group study for GPAT as well as for NIPER. So what we did, that we made a chart, like if we take a root, so cork, cambium, phloem, xylem, we just wrote the important parts which are present in the root. So whichever part is present in that particular root or that particular drug, we just made a tick mark. If it has some special features or any essential things, like if it has calcium oxalate crystal, what sort of calcium oxalate crystal it has, so we had written that in bracket. So both the study is done side by side. Again, in case of cognosy, I would say Pearson has some charts regarding stomata, calcium oxalate, and trichomes, which is very important. Like basic questions are asked, like which leaves has which sort of stomata, or which fruit has which sort of trichomes. So that is very important, and this small points can give you good scoring when you study about cognosy, because cognosy is like, when we study for theory, it's really lengthy because we have to study, not only study its biological source and family, but also its uses, its chemical structure, everything. But in case of GPAT, you all need to know its biological source for sure. But this stomata, calcium oxalate, trichomes, etc. So these things are important, and very few drugs are given in those chart. And as per as my knowledge, or how many question papers I have seen, only those drugs are, only selective drugs are asked in the questions. So when you all do that charts, so that will be really helpful for you all. Next thing in cognosy would be the test in case of like chemical test, different chemical tests are there. Like I would tell example is bond Rager test. As we all know that bond Rager test is done for anthraquinone glycosides, but modified bond Rager test is done for C glycosides. So the difference between two is, I think, only in modified bond Rager test FeCl3 and HCl is used, whether in bond Tracker test only H2SO4 is used. FeCl3 is not used. So these basic questions are there regarding what reagents are used in particular test. So that is very important when we study about test, because they ask that if you want to determine particular drug, what sort of test you use, or which is the test used for determination of amino acids. So ninhydrin test is used for amino acids. So such questions can be very scoring. Next is also biosynthesis. Biosynthesis, there's one more chart in Pearson, which, for example, I would give, they ask the question like what is the origin or what is the source of reserpine or biosynthetic pathway from which reserpine is made. So reserpine comes under indole alkaloids. So indoles are prepared from tryptophan. So what is the source of reserpine? It is indirectly tryptophan. So this basic biosynthetic pathway, there is one chart in Pearson, so also that chart is important. Next topic I would take, that is Sutix. Actually Sutix, I would say that it is really vast, and it is I would say the core of GPAT also, and also core of NIPER, because it includes almost 50% of whole syllabus of GPAT. Each one can have a different opinion. That was my opinion, that mostly questions come from Surtics section. So in Surtics, there are different parts in Surtics. So when we come, I would first start about with physical pharmacy. So in physical pharmacy, chapters like surface tension, rheology are really very important, because there are various questions asked based on the rheology. Surface tension, I would say that look at the formulas, by not by heart those, but clear your concept regarding surface tension. Rheology also, they ask different examples of suspension, that magnesium magma, what is what sort of flow does magnesium magma shows. So such questions are asked. So rheology is really important from PP. Next is powder characteristics. When we say about powder characteristic, I would say that various tables like Cars index, Hosner's ratio, these charts are really important, because they ask that if the Hosner's ratio is this much, then what is the flow of powder? So those questions are really very scoring, and if you all know the chart, then you can solve these questions really fast. Next is also about sieve number. So they ask you that which is the most coarse sieve, or which is the most fine sieve, or what is the sieve number that has dash dash, what I would say, so I think square something it is there, its sieve number has that unit. So that can be asked. Then about more about PP, I would say just focus on rheology. Rheology is really important. Next, going towards Sutix. Sutix basic is tablet, capsule, and parenterals, because it covers major part of GPAT as well as NIPER. In tablet, also biphasic and semi-solids are equally important. Also, aerosols are important, but more questions are asked on tablet, capsule, and parenterals. So in tablet, what to be focused is excipients. Like in Lachman, there are various charts regarding fillers, binders, and other excipients, like what more is there? Fillers, binders, diluents. So such things can be. They have given there more. What I would say, there are generic name and also the proper company name, like Avicel. Avicel is another name for microcrystalline cellulose. But they ask that what is the composition of Avicel. So if you know that chart, then it will be very easy for you to solve such sort of questions. Also, another example would be DIPAC. So what is the composition of DIPAC? It is sucrose and dextrine. So if you know this mostly use names, so you can solve this question. More example would be sugar tab is also used, and Avicel, and many more things are used. I can't recall it right now, but yeah, it's there. In case of capsule, I would say capsule size, there's a chart even in yeah, there's a chart in Lachman also. Banker is very good for capsule. In Banker there is chart of volume fill of each size of capsule. So also they ask that capsule 000, what is the volume fill size of such sort of capsules. So these questions can be easily known. I don't suggest you all to buy hard those fill volume, but at least know what is the sequence, what is the difference. Like if it is that, what is the difference? Like, basically, you should know that in what range if one capsule comes. Like you don't have to buy hard the exact number, but at least you should have an idea about what range the particular size number can come under. Then, as I said, the another names like Echo Gel or Echo Fill. So this they ask that what is the meaning of this. So Echo Gel is used for powders which are filled in soft gelatin capsule, whereas Echo Fill is an short name, or I would say aquafil is used for powders which are filled in hard gelatin capsules. So this sort of words you all should know, and basic points are asked. Nobody asked you certain theories and what I would say, what we study more for our semester, like some theories we have to explain, some diagrams we have to draw. Nobody asks you that. You should know the basic concept and some important words which are consistently asked or most probably used in company. So such things are very important. In case of tablet and capsule, um, QC testing is very important. In QC testing, you have to know like hardness, tablet thickness, then weight variation in terms of USP, in terms of IP. Those charts are given in IP. Also, you'll get this chart in Pearson regarding QC testing, which is very good. I would say just refer those IPQC testing from Pearson. Also, the defects of tablet and capsule are very important because not only the naming of defect or the meaning of those defect, also you all should know the cause and the remedy of those defect, because they ask that if there is a sticking problem in certain tablet, what will you use or what is the remedy. So or they ask the cause. So if you all know these basic things, it will be very easy, and there's nothing much difficult. It's a basic common sense. Like if there's sticking, then why sticking will be cause more binder is there, or drying is not proper. So that we have to just apply your common sense while solving questions and also while doing the study. Because I would suggest that hard study, yeah, hard study is important to give y'all more marks or more scoring. But with smart study, y'all can, in short time, y'all can just qualify GPAT. Because what I did was, I would say it was smart study, because I just studied for 20 to 25 days and I qualified. But I won't suggest you all this, because my score is not that good. So if you all want to do hard study, yeah, you all can refer Piyush, refer Pearson, you all can do that. But if you want to do smart study, just remove your notes and just follow the points which I have told right now. Because what I am telling, I think with this much you all can easily qualify GPAT. But for scoring good, you all need to study much. So these are the points. Yeah, I have to give this disclaimer that this is enough for qualifying, but for scoring you all have to study much. So next, what I was saying about is defects. That defects you all will also get in Banker and also in Pearson. Next is about dissolution apparatus. Dissolution apparatus are different according to USP, BP, and IP. Just remember which type of apparatus is used in which sort of official books, because they ask that what is USP apparatus 1, what is IP apparatus 1. So you all should know the difference between the different apparatus used. Also, for I think tablet weight variation or capsule weight variation, there are different values for IP and different values for USP. So this difference you all should know while going for exam. Next, I would tell about parenterals. Parenterals, I think you all have for this. I mean, last semester, which is eighth semester. In parenterals, okay, you all don't have that thorough knowledge because it is not there for your syllabus right now. But what you should do is different basic difference between water for injection, sterile water for injection. So this is and distilled water. There's one chart in Pearson which gives a clear idea about what is the difference between these different types of excipients used or vehicles used for parenterals. Also in parenteral, important is storage criteria. Also the temperature, various temperature zones, that is very important because they ask that which in which temperature zone does Africa comes, or which temperature zone does India comes. So such questions are there. If you study one chart, that chart is enough for you all to answer such sort of questions. Then in parenterals also, yeah, QC testing is important in case of parenterals also. In case of semi-solids, I would suggest that semi-solids important are bases. like what ointment bases, which are different ointment bases, or which are what is the classification, like aqueous, non-aqueous base, then which are hydrocarbon base. So they ask the example like what is, which among the following is hydrocarbon base. So that if you all know the basic classification of different bases, so it can be very helpful for you all. In case of surfactant, I would suggest thoroughly study the HLB scale, because they ask that what under what HLB the wetting agent comes. So it is 6 to 7. I think, yeah, 6 to 7 or 6 to 8. There's something, there's a range. But that range should be thoroughly known by y'all, because HLB scale is very important. Also, not only HLB scale, but different surfactant have different HLB values, and in Sutex we had different formulas for calculating HLB values. Those formulas can also be seen in Pearson, also in Piyush. Whichever you all want to refer, you all can refer. Just solve different questions about calculating the HLB value of certain surfactants. So there are different formulas. Also, I would suggest that in Pearson, there's a chart regarding names of twin 20, twin 40, twin 60, twin 80, because they can ask you what are this full form of twin 20, or span, what is the full form of span. So it can be asked, so what is the composition. So this question should be known. It is also given in Piyush and Pearson. You can refer any of these books. In case of aerosols, I would say that in aerosols, naming of what I would say, chlorofluorocarbons or CHF, that numbering or naming of CHF is important, because they give a name, or they give a number of what I would say, hydrocarbons, and they tell you to number them or name them. So there's that formula I remember, like C minus 1, H plus 1, and F for naming of CHF. So that formula is very important, because they ask certain questions. Then in aerosols, there are different parts like ferrule, stem, gasket. So this different parts, you all should know the working or what is the use of such part, and also what should be the composition, or what is the composition, or what it they are made of in aerosols. Like I would give you example, ferrule is made up of aluminum or tin. Then stem is made up of nylon or derylene, and gasket is made up of rubbers, different type of rubbers like buna and etc. So there are simple questions. I think more questions in NIPER regarding different functions of aerosols and the composition. I had not seen questions regarding aerosols in GPAT, but yeah, there are questions regarding aerosols in NIPER. Then next in Sutex, I would go with posology. Posology, it is actually very vast. I won't say vast, but it's very confusing, because there are various number of problems, and not each and every problem can be solved by certain method. So you all need to be thorough with the formulas. If you all just what I would suggest, basically, if you want to do posology, I would suggest do the dose formulas like Clerk's formula, Fry's formula. Those formulas are used to calculate the child dose. So these formulas can be asked in posology. Also, there are various conversions like minim to drop or kilos, etc. This, what is 1 teaspoon full, what is the volume of 1 teaspoon full, what is volume of 1 tablespoon. So these questions can be asked regarding posology. So if you all are good with maths, you can for sure go for this, because it is also very scoring. I think one question was asked also for us in NIPER, maybe for dose calculation. Next, I would tell about jurisprudence. Jurisprudence, I think you all have in seventh semester, and that's a actually I would say advantage because you all are going through this jurisprudence currently in your syllabus. So you all will be well versed with the different schedules. So basically काही केलं नाही ज्युरिस मधलं तरी चालेल पण शेड्यूल्स नक्की करायचे कारण at least there's one question on what comes under particular schedule. So this question is basically asked, or I would say confirm there's one question on schedule because that can give you 4 marks if you just study those schedules from A to Z. um, there are different tricks to study schedules. Like I would give you an example what we did because I think examples make it more easier. So when we see a snake, we just do the E. So we had remember that E means poison. Means this E schedule E comes for poisons used in for poisons used for preparation. Then E1 comes under poisons used for Ayurvedic preparations. So there's difference. Like such examples, you all can by taking such examples, you all can by heart various schedules. I would again give an example like for schedule T, one of our classmate Trupti, she had done her final year project regarding Ayurvedic preparation. So we had just remembered that Trupti, okay, T is equals to Ayurvedic, means factory requirements for Ayurvedic preparations. Like this, you all can name each other or do any sort of tricks, but by heart all the schedules thoroughly. Next, I would say also the appendix. I think six or five appendix are there in Pearson. Yeah, in Pearson, in jurisprudence part, there are different sort of appendix. Only one line sentences are there regarding appendix are based on clinical studies, stages of clinical studies. So that should be known. Again, if you all want to go more deeper in jurisprudence, by heart the form numbers because form numbers they ask about that for what purpose this form number is used, or they can also ask a reward like for obtaining license of certain company. So which form number should be filled? So such questions can be asked in jurisprudence. Next, IPR. I don't know whether you all have selected or not IPR as your subject, but in IPR, what important I would say that is patent copyright. So, and various other things like not recalling it right now, but patent, copyright, etc. These things are there. You all need to know the duration. What is the duration of patent? What is the duration of copyright? What is the duration of geographical something is there. So
And
you
laws related to patent, copyright, there are different laws. So you can remember this.
yeah, also laws are there, different duration is there regarding IPR. So next I would go regarding micro. So in micro, I would suggest you all to do a chart. It is both available in Piyush as well as Pearson regarding different causative organisms for different sort of disease. Also, when we study about vaccines, study like which are live, which are attenuated or live attenuated, which are killed vaccines, or which are obtained from virus, which are obtained from bacteria, which are obtained from toxoids. So make a chart or ready-made chart is already available in Piyush as well as Pearson. Next, important in micro is about immunity. Immunity like innate immunity, acquired immunity. What is basic classification? What are examples of primary immunity, etc. Next is immunoglobulins. You all need to know the function of each immunoglobulin, its duration, where it is, like what is the function, where it is synthesized, what is the percentage, etc. of immunoglobulins. So this is the important topics in micro. Next, I would go with biopharmaceutics. Biopharmaceutics, we are not that versed about biopharmaceutics because it was not much included in our syllabus. But basically, I would suggest that if you want to study biopharmaceutics, go through the theories of different theories of dissolution, then a biopharmaceutical classification, because they ask that which drug, what is the BCS classification of propranolol or etc. Like they give a drug and ask you all which class it comes under. So this chart is also given in Brahmankar, also in Pearson, Piyush. Each book you all will definitely guess this BCS classification. Next is rate kinetics and rate kinetics related problems. Rate kinetics related problems are for sure asked in NIPER as well as GPAT, because these problems are at times very easy or at times really confusing. So see, if you all like to solve problems, you all can for sure go for it because it will give you a good scoring. The problems relating to half-life, renal elimination, also dialysis rate, etc. All these formulas are clearly given in Brahmankar. Just make a chart of these different formulas, which will be really helpful for y'all when y'all do the overall study for GPAT. Like tomorrow is your exam and you all have to revise the formulas. No need to go for hunting of books. Just open that chart and go through all the formulas, because formulas are really easy, but it can be confusing if you all don't revise it, or if you all keep on searching it for the end moment. If you all have a chart ready with you all, it can be really easy. And believe me, these problems are really scoring in both the exams. Next, about engineering, I would say different examples of mixer, blenders, mill. So, what is the use of different mill? Like, what sort of material it can be used? Which mill can be used for which material, or it cannot be used for certain material? There's a chart in Pearson as well as Piyush regarding this meals, mixers, blenders, which is like they ask that which mixer is used for highly viscous fluid, which is used for low viscosity fluid. So if you all know these basic examples, so it can be very easy. In case of cosmetics, there's nothing much to refer, but you all should, as I said, when we study for semi solids, you all can also study for different base used for cosmetics, like what is the base for creams, what is the base for lipstick, etc. Like you all should know the basic component or basic dye, what are different types of dyes, um, different types of flavorants or different type of colorants. Like it was, I think, about question about colorant. I don't remember the exact question, but it was question regarding colorant, that this colorant gives what sort of color. So the such questions are asked if you're in cosmetics. Then this was all about suitics. Now, my suggestion regarding would be Cology and Medchem. So Cology and Medchem, I suggest you all to study side by side, because when we study ecology, we study its classification, like therapeutic classification, and in medichem we study its chemical classification. So if you study both the classification side by side, its therapeutic as well as chemical classification, it will be easy for you all to remember it. If you note down the theoretic classification on this page, on side page you just note down its chemical classification. Also, I would suggest that certain drugs, y'all, they have different sort of adverse effect, or yeah, adverse effect, or they have certain advantage over other drug. That's why they are mostly used. Such drugs, y'all have to just mark or make a note that okay, half life of this drug is this much. Okay, this potency, this drug is more potent than another. Just y'all have to note it down. Also, adverse effect, like adverse effect of phenytoin is, many times that question is used, that what is hirsutism is adverse effect of which drug. We know that okay, it is about phenytoin. So this certain disadvantage or adverse effect, You all have to just note down these important points while studying for Cology and Medchem. Then, going further towards organic chemistry or biochemistry, um, I basically don't like chemistry, or I'm not good enough for guiding you all regarding chemistry. I would suggest that Siddesh or Sayuja, both of them, will give an actual idea about studying chemistry. I would suggest I can tell you more about Sutix, Cology, Cognosy for sure, but not much about organic chemistry and biochemistry, because I personally had not done both of these subjects for GPAT, not at all also for NIPER, because I don't understand anything from that. So I won't tell you all that do like me, because my score is not good enough to keep an ideal. But giving an overall idea in biochem, I would suggest that go through different pathways, what how much ATPs are synthesized from different pathway, that is important, or what is the precursor of certain pathway, which pathway takes place in which part of body, that certain takes place in cytosol, certain takes place in mitochondria. So what is the location of different pathways is important in case of biochemistry. Also in organic chemistry, stereoisomerism is really very important. And according to my experience, I can tell you that for GPAT, you all need to go through like detailed study of whatever we have studied from first year to final year. And for NIPER, it's not that deep, because you all have to just revise whatever you all had studied for GPAT. And NIPER mostly focus on chemistry and stereochemistry, because it gives you direct the structure, and they ask you that what will be the isomer of this, or they give you in structure, and they tell you all to nomenclate whether it is R rotatory or RS configuration, or whether it is dextrorotatory, it is levorotatory, etc. So this stereoisomerism is more important in case of NIPER, but not that much in case of GPAT. Again, so this was all about what I personally studied for GPAT and NIPER. If you all have any questions regarding to that, you all can ask me at the end, if the
Yes,
time
yes, yes, sure. At the end, we will go for the question and answer session. Right now, if you are finished, so better
yeah.
I would like to request Prajakta. You can proceed for your session. Yes, Miss Prajakta. Hello, Prajakta. You're audible. Yes, am I audible to you? Can you hear me? Hello.
Sir, I think she's not there.
Uh, just yes, Mona is there. Mona Mohite. Hello, Mona. Are you there?
Yes, sir.
You can
Yes,
proceed.
sir.
Yes, you can
Hello.
proceed. Okay, we
Uh,
can wait. Okay, after that,
Good
Prajakta.
evening.
Yeah.
Good evening to all the respected teachers and dear juniors. Today I am here with you to share my experience about my GPAT preparation. So, first of all, I want to tell you all that whatever time you had now left with you, that is this 100 days or 3 months. So, try to solve maximum questions about try to solve maximum question. If you are referring Inamdar, means I had referred Inamdar for the GPAT preparation. So if you are doing, if you are trying to solve these questions, then you will. Hello.
Hello. Yes, Mona. You can
So
continue.
I'm so I must suggest you to solve maximum questions so that you can get idea about which type of questions are generally asked and which topics are generally get covered. Means you can get idea which topics which the questions are
Mona,
generally
I will
asked.
suggest you
As
if you are
Pritam
using
already has
headphone.
told.
Mona,
Mona, your voice is breaking.
Yes,
if
sir.
you are using a headphone, then remove the headphone and speak as such. And try to switch on the camera, so I will take the snapshot.
Mona, are you with
Hello,
the good
sir.
good connectivity? Yes. Little bit voice is breaking. Yours.
So sir, connectivity issue is there. Hello, sir. Can you hear me?
Yes, yes. Proceed. Speak up.
So when you solve exam questions, you will come to know that which type of questions are asked and which topics will generally get covered, so that you can study those topics in depth. Pritam has already told the topic which you should study in detail. So if you are ready, uh, so you and when you solve questions, you will be very
Hello, Mona. I think your voice is too much breaking. Better we should ask to Prajakta, and meanwhile you can try to find out the good connectivity. So we should continue. We can continue. Okay, afterwards.
Okay.
Is it okay to
Sir,
you?
yes,
Yes.
sir.
Mona, just
Sir,
meanwhile you can try to connect yourself with the good
she is
connectivity.
trying to say you have to solve maximum question paper. That
Okay.
is, at least we used to solve a 5 to 10 question paper, minimum 5 to 10 question paper. And while solving question paper, for example, there is one question, which of the following is directly acting skeletal muscle relaxant? Four options are there: dantrolene, tuberculin, pancurium, and succinylcholine. So the answer is dantrolene. So dantrolene, it is directly acting skeletal muscle relaxant, and other three are indirectly acting skeletal muscle relaxant. So while solving question paper, try to figure out why other three options are wrong, and try to read related to that question. That is, for example, in case of skeletal muscle, so what is the classification of skeletal muscle relaxant? What is mechanism of action of skeletal muscle relaxant? What is the duration of action? And try to study special drugs like tuberculin. So when you solve question paper like this, no, your half of study will done automatically.
Hello, Prajakta.
She is having network issue, I guess.
Issues, I think she is there, but we are not able to hear her sound. Okay, it is not reachable. Yes, Prajakta, can you hear? Hello. Yes, Mona, are you there? Mona. Hello, Mona.
So what she was, shall I continue?
Yes, yes, yes.
So
Once she will join now, then we'll continue it. Okay. Yes, you can proceed.
what she was going to tell everyone about what is important in group study and how you all should prepare actually for GPAT as well as NIPER. So while doing group study, discussion is really very important, and group study is actually very beneficial because in minimum time you all can cover maximum syllabus. As I gave an example about cognizy, like I studied roots, she studied for fruits, Akshata studied for leaves, and next day we would just discuss about, okay, these points are there in roots. I would tell them that, okay, this drug comes under roots, this is the biological source, these are certain main points in this drug. They would tell me about their part, so even I am revising my part of what I had studied, and also I am able to understand what they had studied. So we have consumed our, or we have
Time.
conserved our time.
You can do
Yeah.
Yes, you can
maximum work
continue.
in minimum time.
Yes, Prajakta.
So what are you trying to say, Pritam, about group discussion? No.
Yeah.
Yeah. Okay. Secondly, without taking any tension, you can enjoy the process of preparation. Like if म्हणजे तुम्हाला खूप टेन्शन येते की यार किती
Mhm.
अभ्यास करायचा आहे वगैरे, so instead of taking tension, you can start enjoying this process while doing group discussion. So I should suggest go with this method, because I personally experienced better result of this method. I used to study for each and every semester for NIPER, and for GPAT also we did group discussion. So other thing I want to tell you guys, your competition is not in between your class, it's in between 4 to 3,000 students which are competing from different state. So guys, your YBCOP rank is not going to display, your all India rank is going to display. So please help each other, don't be hesitate to share your notes to others, try to help each other. So by this you can save your time, and in short time you can do extra work. So the next point is, when
अरे यार.
Hello. Might she lost her connectivity. Pritam?
Yes, sir.
Might be, but she is there. I think in the, yes, I can see her name in the meeting as a participant.
Next, she wanted to share
Huh.
about what is the number of questions. What we insist you all, like we had solved 70 to 80 questions or 60 to 70 questions during GPAT, because what we noticed while practicing question papers, like आमचे तुक्के लागत होते. आम्हाला माहित होतं की ओके बाबा, ह्या तिघांमध्ये नसेल, हे तरी असणार, किंवा तुक्के लावण्याची एक टेक्निक असते मोस्टली जीपॅट मध्ये. If you all don't know, the longest sentence which is there in the options is actually
is
the answer
correct
to the
answer.
question.
Yes.
Yes.
That is the most known technique about तुक्के लावणे. तो कुठेतरी ऐकलेलं असतं, कोणीतरी हे बोललेलं असतं, आपल्याला ते आठवतं किंवा ग्रुप डिस्कशन मध्ये, हा बाबा हिने ह्याच्यावर जोक मारलेला. सो असे काही थिंग्स असतात. तुक्के ब्लाइंडली कधीच लावायचे नसतात. अशा क्वेश्चन्सचे तुक्के लावायचे तुम्हाला थोडा तरी अंदाज असेल की ओके हे ऐकलंय किंवा हे मी वाचलंय. किंवा मोस्टली ब्लाइंडली सांगेन तर हा सर्वात लॉंगेस्ट जे क्वेश्चन किंवा लॉंगेस्ट सेंटेन्स असतो तो मोस्टली आन्सर असतो. हे आमचा एक्सपिरियन्स होता आणि आतापर्यंत तर तो बरोबर आलाय. कसं
Oh,
कधी चुकीला बघितला नाहीये.
Related with that question and related with that.
या.
Yeah, this is a one kind of trick I can miss. You can, or you would like to suggest, or we can say you would like to share with your juniors. This is a one of the we can say practice by which they can qualify for. Yes, continue, Prajakta. Yes.
yeah. And in case of NIPER, besides studying,
Hello, Pritam.
या.
I think better way we would suggest to Prajakta. She can put her video off and she can continue. That will be a more better.
ओके.
Pritam, hello.
You can put your video off and you can continue. That will be a more better, I think, because
But sir,
video. Yes. Sorry.
I really have some network issues, so
Okay. Okay. Yes, we can
it is
understand your
better to.
problems.
So sir, can Pritam will cover my part? It's
Yeah. Sure. Sure.
better.
No problem. Yes.
ओके.
Pritam, you can continue.
Was trying to say that in NIPER there are more questions regarding mathematics. Like I won't say more questions, but there are at least 8 to 10 questions regarding mathematics, basic mathematics. What is simple interest? What is complex interest? What are the formulas of these questions? Again, what we studied during our fourth and eighth standard scholarship, like what is the relation of this person with that person. एक क्वेश्चन होता नायपर मध्ये आम्हाला, लाईक person named something Seema is standing on fifth position from left hand side, and something is there XY person is standing from with this position number from right hand side. If they interchange their positions, what will be the difference between the two of them, or from which position will that person will be from right hand side. So these basic questions are there where you all can easily solve those. I would also like to give another example, like हाईट दिलेली होती 10 मुलांची, की ओके ह्याचं ह्याची हाईट ही आहे, ह्याची हाईट ही आहे, 10 मुलांची हाईट एवढी आहे, 5 मुलांची हाईट एवढी आहे. सो व्हॉट इज इफ टोटल नंबर ऑफ स्टुडंट इन क्लास आर 30, देन व्हॉट इज द एव्हरेज हाईट ऑफ स्टुडंट्स इन दॅट क्लास. So this sort of questions are asked in NIPER. These questions are not asked in GPAT. Also in NIPER, I would say questions like who is the CEO of Cipla, or what more questions like who is the author of certain book. They are out of box questions. There are 8 to 10 questions definitely on certain things like who is the author of this book, or they give us a box and you have to find out that which is the odd number which between these numbers. Then also they ask different odd man outs. Certain questions are really very easy, like y'all can just see that question and y'all can answer it, but certain questions have an difficulty level. And in case of Niper, y'all have to solve 200 questions only in 2 hours. So what problem I faced was, I was not able to manage time. That's why I could only solve only 120 questions, whether where my target was about to solve 140 to 150 questions, where because नायपरमध्ये तुम्ही रिस्क घेऊ शकता, कारण +1 आहे, -0.25 आहे. जरी चुकला तरी 0.25 मायनस होणार आहे. आणि त्याच्यामुळे काय होणार, तुमचं जीपॅटला कसं होतं, +4 असतं, -1 असतो, त्यामुळे स्कोअरमध्ये ना खूप मोठा डिफरन्स येतो. नायपरला 0.25 0.50 असा स्कोअर चेंज होणार तुमचा जर क्वेश्चन चुकले तर. सो मोर रिस्क घ्या तुम्ही. मोस्टली तुक्के लागतात तुमचे तर बिंदास तुक्के लावा नायपरला. बट जीपॅटला तसं नाही करायचं. जीपॅटला तुम्ही बघा तुमचे तुक्के लागतायत का, क्वेश्चन पेपर सॉल्व्ह करताना किती तुम्हाला क्वेश्चन पेपर इझी जातोय किंवा टफ जातोय, त्याच्यावर तुम्ही क्वेश्चन नंबर किती अटेम्प्ट करायचे हे डिसाईड करा. बेअरली लाईक आम्ही जसं डिसाईड केलं की 70 to 80, ओके आम्ही डिसाईड केलेलं 70 to 80 सॉल्व्ह करायचे, पण तिथे जाऊन काय झालं की मला स्टार्टलाच पहिले केमिस्ट्रीचे क्वेश्चन आले. केमिस्ट्री आल्यानंतर मी थोड्या वेळासाठी घाबरली होती, कारण मला केमिस्ट्री काही येत नाही. सो असं काही असेल ना, तेव्हा मी ठरवलं की ओके बाबा 40-50 पर्यंत मी जाईन, जास्त जाणार नाही. बट नंतर जेव्हा मी सॉल्व्ह करत गेली तेव्हा कळलं की ओके बाबा येत आहेत मला क्वेश्चन. सो म्हणून मी आय थिंक 79 की 78 असे क्वेश्चन सॉल्व्ह केले होते जीपॅटला, आणि नायपरला 120 केले होते. जीपॅटला तुम्हाला 125 क्वेश्चन्स आहेत पण 3 तास आहेत, पण नायपरला 2च तास आहेत आणि 200 क्वेश्चन्स, 2 तास आहेत आणि 200 क्वेश्चन्स आहेत. त्यामुळे तुम्हाला खूप फास्ट सॉल्व्ह करायला लागणार नायपरला. आणि जीपॅटला तुम्हाला सॉल्व्ह करून फुल क्वेश्चन पेपर ऑलमोस्ट आवर टू हाफ एन आवर असतो. परत स्वतःचा पूर्ण क्वेश्चन पेपर चेक करायला अँड पूर्ण सगळं रिवाईज करायला. आय थिंक बऱ्याच जणांनी थर्ड इयरवाल्यांनी एक्झाम दिली होती जीपॅटची, तुम्हाला पॅटर्न माहिती आहे. भलेही त्यांनी जीपॅटला असं दिलेली असते की ओके मेडकेमचे एवढे क्वेश्चन, सुटिक्सचे एवढे क्वेश्चन्स, बट प्रत्येक सेक्शनमध्ये क्वेश्चन मिक्स असतात. सुटिक्समध्ये फक्त सुटिक्सचे क्वेश्चन्स नसतात, मेडकेमचे पण क्वेश्चन असतात, कुठे कॉग्नसीचे पण असतात, अदरमध्ये मोस्टली केमिस्ट्रीज असतं. आमच्या इथं तसंच होतं, मला तरी तसं आलेलं. सो असं ठरवून ठेवू नका की ह्याच्यातले एवढेच सोडवणार, त्याच्यातले तेवढेच सोडवणार. तेव्हा क्वेश्चन पेपर बघून तुमची काय कंडिशन असेल, तुम्हाला किती क्वेश्चन्स येत आहेत, त्याच्यावर डिसाईड करा की काय सॉल्व्ह करायचं आहे. आय वुड परत सजेस्ट करेन की ओके तिथे काउंटिंग सिस्टीम असते की तुम्ही किती क्वेश्चन्स अटेम्प्ट केले, किती सोडले आहेत, किती ना रिव्ह्यू दिलाय. बट मी पेपरवर ओके आपली ती स्टॅटिस्टिकची सिस्टीम असते, 1 2 3 4 आणि 5ला आपण डॅश करतो. असं मी करत होते. ओके बाबा हे एवढे क्वेश्चन्स माझे कन्फर्म आहेत, मला माहिती होतं की ह्याचं आन्सर बरोबरच असणार, हे चुकणार नाही आहे. एवढे क्वेश्चन्स मी केले होते. ते माझे 40 to 45 असे क्वेश्चन्स होते जे मला माहिती होतं कन्फर्म हे बरोबर असणार आहेत. त्यात अंदाज चुकला ही वेगळी गोष्ट, बट ॲटलिस्ट एवढे क्वेश्चन्स ठेवा की तुम्हाला 250च्या वरती स्कोर मिळेल तुमच्या कन्फर्म क्वेश्चन्सवर. बाकी तुम्ही तुक्के लावा, थोडंसं वाटतंय ऐकलंय, असे अनसर्टन क्वेश्चन असतात ते 50-50 ठेवायचे. चुकला तर ओके -1 होईल, बरोबर आला तर +4 मिळून जातील. कुठेतरी बाकीचे चुकलेले जे आपल्याला वाटतायत कन्फर्म आहेत तिथे हे मार्क्स ॲडजस्ट होतील. बट कोणाच्या सांगण्यावरनं जाऊ नका की किती क्वेश्चन सॉल्व्ह करायचे. तुम्हाला त्यावेळी काय वाटतंय तेच करा. आणि 80 किंवा 90 क्वेश्चन्स मॅक्स टू मॅक्स ह्याच्यावर जाऊच नका, आय वुड सजेस्ट. कारण आपल्याला वाटतं हा बाबा हे बरोबर आहे, पण तसं नसतं. कारण त्यात खूप लूप होल्स असतात आणि जीपॅटचे ऑप्शन्स खूप कन्फ्युजिंग असतात ॲज कम्पेअर्ड टू नायपर. नायपरचे स्ट्रेट फॉरवर्ड क्वेश्चन होते, बघितल्यावर समजत होतं ओके बाबा हे आन्सर आहे. आणि आम्हाला तरी 2 ते 3 क्वेश्चन्स रिपीटेड होते आमच्याच क्वेश्चन पेपरमध्ये इसेन्शियल अमिनो ऍसिड बद्दल, की हा कोणता इसेन्शियल अमिनो ऍसिड आहे, कुठला नॉन इसेन्शियल अमिनो ऍसिड आहे. हा एकच क्वेश्चन आम्हाला 2 ते 3 वेळा विचारलेला होता. सो नायपरला सुपरफिशियल स्टडी आहे, जीपॅटचं रिव्हिजन आहे, बट जीपॅटला तुम्हाला डिटेल स्टडी करायला लागणार आहे. नेक्स्ट प्राजक्त अजून काय राहिलंय?
Hello. I think Prajakta is not there. I think she left. Better. Yes, you should continue. Otherwise, Mona, she is available. Okay.
मोना
Mona, are you there? Hello, Mona. You should continue. Yes.
हॅलो
Yes, you can continue. Yes, you
हॅलो
should.
सर, आवाज येतोय?
Yes, speak up.
हा,
Yes,
जसं
continue.
प्रीतम मला म्हणाले की कॉन्फिडन्स लूज करू नका स्वतःचा. जेव्हा आपण क्वेश्चन आन्सर सॉल्व्ह करत जातो, कधी कधी असं होतं की आपले क्वेश्चन चुकत जातात आणि आपला कॉन्फिडन्स लूज होत जातो की मला काही येत नाहीये किंवा जमत नाहीये. पण कॉन्फिडन्स लूज करू नका. जेवढा वेळ आहे त्या वेळात क्वेश्चन वाचा. जर इनामदार रेफर करत असाल तर मागे एक्सप्लेनेशन पण दिलेलं असतं त्या आन्सरचं, की हे आन्सर का आहे, बाकीचे आन्सर्स का नाहीयेत. तर ते एक्सप्लेनेशन रीड कराल ना, तेवढं खूप चांगलं आहे, कारण आपल्या मॅक्झिमम कॉन्सेप्ट त्यातूनच क्लिअर होत जातात. आणि फायनल जीपॅट जो आहे ना, तो खरंच ते त्या एक्झामच्या वेळेला आपला प्रेझेन्स ऑफ माइंडवर डिपेंड आहे. म्हणजे जर तुम्ही मॅक्झिमम क्वेश्चन सॉल्व्ह केले तर असं नाही की स्कोअर चांगलाच येईल. जर तुम्ही पेपर झाल्यानंतर जेव्हा उरतो तेव्हा स्वतःचे मार्क्स टोटल करून बघू शकता. जर तुम्हाला वाटत असेल की हा माझा स्कोअर तेवढा अचीव्ह होतोय की मी क्वालिफाय करू शकतो, तर तुम्हाला गरज नाहीये की अननेसेसरी असे क्वेश्चन सॉल्व्ह करा ज्याच्याबद्दल तुम्ही डाऊटमध्ये ज्याच्या शुअर नाही आहात. तर गरजेचं नाहीये की असे क्वेश्चन सॉल्व्ह करा. आणि जर एखाद्याला वाटतं की नाही मी अजून माझा स्कोअर तेवढा वाटत नाहीये होईल असा, तर ते रिस्क घेऊ शकता. तर तुम्ही मॅक्झिमम क्वेश्चन सॉल्व्ह करू शकता, जेणेकरून तुमचे जर तुक्के लागलेच म्हणजे प्रोबॅबल आन्सर असले आणि ते बरोबर आलेच तर स्कोर तुमचा वाढू शकतो. सो त्याबद्दल आताच डिस्कस करणार नाही. आता जेवढा वेळ तुमच्याकडे आहे, तुम्ही मॅक्झिमम क्वेश्चन सॉल्व्ह करा. खरंच मग हेल्पफुल होईल. बस, दॅट्स ऑल.
Okay, thank you. Again, I would like to, or we would like to hear from on the behalf of students. Okay, which you can just better you should suggest to your juniors which book they have to refer for preparations, or we can say some study as a study material. So that would be a more beneficial to your juniors. Yes, please. And then we will go for question answer session. Yes,
Oh,
Mona,
uh,
Pritam, and yeah, one by
so
one.
regarding books, I would say Piyush, Pearson, Inamdar, these 3 books are really good. First, if you all have to second year पासून प्रिपेअर करताय, पियुष घ्या. पियुष मध्ये थरर दिलाय तुम्हाला, लायब्ररीमध्ये जाऊन वेगवेगळे बुक्स रेफर करायची गरज नाही. पियुष मध्ये एकत्र तुम्हाला सगळं दिलेलं आहे. तुम्ही थर्ड इयर मध्ये आमच्यासारखं अगदी लास्ट मंथ मध्ये प्रिपेअर करताय, आय वुड सजेस्ट की पिअर्सन वाचा. पिअर्सन जरी तुम्ही कम्प्लीट वाचलं तरी पण बेस्ट आहे. अगदीच नाही त्यात पण तुम्ही जर शेवटच्या 15 दिवसात प्रिपेअर करताय तर तुम्ही इनामदार वाचा. इनामदार मध्ये क्वेश्चन पेपर दिलेत आणि प्रॉपर एक्सप्लेनेशन आहे की ओके हे आन्सर आहे तर का आहे, हे 3 आहेत तर हे का 3 नाही आहेत. त्यामुळे खूप कन्सेप्ट क्लिअर होतात शॉर्ट टाइम मध्ये. त्यामुळे त्या फेज मध्ये तुम्ही इनामदार वाचा. इव्हन तुम्ही सेकंड इयर पासून स्टडी करताय, ओके पियुष आधी संपवा, मग पिअर्सन वाचा, मग इनामदार वाचा. देन क्वेश्चन पेपर पियुष मध्ये खूप जास्त दिलेले आहेत, त्यामुळे खूप प्रॅक्टिस होते. पिअर्सन मध्ये पण आहेत बट कमी आहेत. अँड इनामदार तर कम्प्लीट क्वेश्चन पेपरनेच भरलेलं आहे. सो प्रत्येकाची स्टडी मेथड वेगळी असते की तुम्हाला नोट्स काढायचे आहेत की डायरेक्ट क्वेश्चन सॉल्व्ह करायचे आहेत. सो अकॉर्डिंग टू युअर फिजिबिलिटी, बेसिक बुक्स आय थिंक मी जसं सांगितलं की आपले निराली प्रकाशनचे जे बुक्स आहेत ना, कोकारे बोला किंवा इंजिनिअरिंगसाठी ते ग्रीन आणि ब्लू कलरचं 2-2 बुक्स आहेत, ते बेसिक बुक्स आहेत. इव्हन सुटेक्सचे पण ते छोटे छोटे बुक्स आहेत ना, त्यामध्ये खूप शॉर्टमध्ये आणि इसेन्शियल इन्फॉर्मेशन दिली आहे. त्यामुळे जीपॅटसाठी नोट्स काढत असताना ही बेसिक बुक वाचायची रादर देन फोय विल्सन घेऊन बसण्यापेक्षा, रेमिंग्टन, लॅकमन घेऊन बसण्यापेक्षा ना, ही छोटी बुक्स घेऊन बसलात ना तर तुमचा कमी टाइम मध्ये खूप जास्त सिलेबस कव्हर होणार. हे असा माझा तरी एक्सपिरियन्स आहे. इवन स्पेक्ट्रम अकॅडमीचं जे लास्ट इयर आमच्यासाठी सेमिनार झाला होता त्यामध्ये पण त्यांनी हेच बोलले की आधी बेसिक बुक्स वाचा मग मोठ्या बुक्सकडे जा. बट मोठी बुक्स वाचण्या एवढा आमच्याकडे वेळ पण नव्हता. ऍक्च्युली आम्ही तो युफोरिया आणि स्पोर्ट्स त्यामध्येच खूप जास्त इन्व्हॉल्वमेंट असल्यामुळे तेवढा आम्हाला इन्व्हेस्ट पण नाही करता आला. तसं आमच्यासारखं तुम्ही करू नका. तुमच्याकडे ऍक्च्युली खूप टाइम आहे. करूनच सगळं चालू आहे. तो हा टाइम ऍक्च्युली चांगल्या ह्याच्यात घालवा. तुमच्याकडे आता बुक्स नसतील म्हणजे लायब्ररीत तुम्हाला जाऊन एक्सेस करता येत नसेल तर पियुष घ्या. फॉर शुअर त्याच्यामध्ये एकत्र सगळं दिलेलं आहे. जर पियुष नसेल घ्यायचं तुम्हाला तर एकाने कोणीतरी लायब्ररीमध्ये जावा, हे सगळी बेसिक बुक्स घ्या, त्याचं PDF करा किंवा ऑनलाईन अवेलेबल असेल तर डाउनलोड करा, नोट्स काढा. बट ग्रुप स्टडी करा, बुक्स रेफर कराच, बट स्वतःचे नोट्स हे खूप इम्पॉर्टंट आहे. किंवा किंवा आदल्या दिवशी तुम्ही जाता ना तेव्हा जेव्हा स्वतःचे नोट्स बघतात की ओके बाबा मी एवढं केलंय, एवढं माझी प्रिपरेशन आहे, तो एक वेगळ्या लेवलचा कॉन्फिडन्स असतो की माझी प्रिपरेशन झाली आहे. ओके, खूप भीती वाटते. माझ्या तर पोटात गोळा आलेला जेव्हा मी सेंटरच्या बाहेर उभी होती. माझे हात थंड पडलेले पूर्णपणे. बट एकदा बॅगकडे बघितलं, ओके बाबा त्यात माझे नोट्स आहेत, मी एवढं प्रिपेअर केलंय, सोबत प्राजक्ता पण होती, मग तिने थोडासा कॉन्फिडन्स दिला. सो आपण जेव्हा ग्रुप स्टडी करतो तेव्हा ते पण एक इम्पॉर्टंट असतो की आपला फ्रेंड आपल्या सपोर्टसाठी असतो की एक कॉन्फिडन्स. ओके आपण घाबरलो तर निदान तो असतो की ओके हा आपण केलंय बाबा, तू घाबरू नको, आपण काही होईल, ओके आपण क्वालिफाय होतोय. सो दॅट इज व्हेरी इम्पॉर्टंट. कारण तुम्ही भलेही किती प्रिपरेशन करा पण त्या मोमेंटला जेव्हा एक्झामला बसणार तेव्हा तुमचा माइंडसेट काय आहे, तुमचं कॉन्फिडन्स काय आहे ना, यावर खूप काही गोष्टी अवलंबून असतात. जेव्हा आपण हॅपी असतो ना आपले इझिली तुक्के लागतात. बघा तुम्ही कधी एक्सपिरियन्स करा. आणि जेव्हा आपण टेन्शन मध्ये असतो ना आपले तुक्के कधीच नाही लागत. सो जेवढे रिलीफ राहाल, जेवढे हॅपी राहाल ना तेवढं चांगलं आहे. लास्ट मोमेंटला अजिबात टेन्शन घेऊ नका. हे मी तरी सजेस्ट करेन. ओव्हर टू
Okay.
मोना.
Yes, Mona, would you like to add? Uh, yes, anything
अ,
else regarding this?
हा, मी तरी खरंच प्रिपेअर केलं होतं. म्हणजे माझी पकड अशी होती, की मी आन्सर क्वेश्चन वाचायची, त्याचा आन्सर मागे आणि एक्सप्लेनेशन आणि त्यानुसार मी ते एक्सप्लेनेशनवरच म्हणजे फोकस जास्त केलेला आणि तिथूनच स्टडी केली होती. तरी सफिशियंट आहे जर तुम्ही
Oh,
केलं तर.
okay. Yes, Prajakta, over to you. Would you like to add anything else? Any more? Yes, Prajakta. She is there. Yes, Prajakta. Yes. Hello. Uh, I think she is not able to hear. Yes. Yes, Pritam, Mona, better way we should go for the question and answer session. Okay. Yes, those who are the participants, dear students, those who want to ask anything regarding the GPAT preparation as well as the NIPER preparations, yes, you can ask. Okay, this is a we can say what interactive session. You can ask. Okay, don't be hesitant. Be frank. Yes. Hello. Anybody would like? Yes. Would you like to ask any questions, students, dear students, participants? Hello. No one is. Yes. Hello. Yes, from the final year students. Anybody else? Third year students? Anybody?
हॅलो
Yes, Dinesh.
सर दिनेश.
Yes.
तुमच्याजवळ जर PDF वगैरे असतील ना इनामदारच्या म्हणजे जर PDF असेल बुकची तर मुलींजवळ वगैरे शेअर केला तरी चालेल म्हणजे आमच्या क्लासमध्ये तेवढी पोहोचेल ती म्हणजे तुमच्याजवळ कोणाजवळ असेल तर.
Yes, exactly. This is a good. Okay, Miss. At least you should share with your juniors, so that will be really helpful for the GPAT or NIPER aspirants. Yes, over to you,
आमच्याकडे
Prajakta.
आउटबुक होतं इनामदारचं आणि पियुषचं. पण पिअर्सन पण एका दोघांकडे होतं. पिअर्सनची सॉफ्ट कॉपी आमच्याकडे आहे, ती आम्ही ज्युनियर्सला ऑलरेडी फॉरवर्ड केली. आय थिंक ओंकार दिवेकर वगैरे त्यांच्याकडे ऑलरेडी
नाही
आहे.
पिअर्सनची आहे फक्त
नाही
इनामदारची नाही त्यामुळे
इनामदारची आणि पियुषची अवेलेबल नाहीये आमच्या जवळ पण.
होतं, त्यामुळे मग आम्ही सर्च पण नाही केलेली.
हा.
बघा
And
तुमच्याकडे ज्यांच्याकडे असेल त्यांच्याकडनं PDF वगैरे जमत असेल तर बघा.
anybody have a Piyush publications, because I referred at my time Piyush publications only book for my GPAT preparation
I
or
think
GATE.
about Inamdar I had shared on the group,
Yes, sir. Yes, I
GPAT
know.
group.
So
It's
I think
available
Mode sir shared this book in Amdar.
last month only.
But sir, I think that link is not open. I think because of I don't know
I had
why.
downloaded. Once again, I will share that.
Okay, sir. Yes. Anything else? Anybody would you like to ask any questions? कोणाला काही questions विचारायचे आहेत का, dear participants, students? Especially we are expecting the questions from final year. Yes. Anybody else? Final year students. Okay. Within 2 or 3 months, you have to appear for GPAT examination. Yes. Hello, Mr. Omkar, Hina, Asmita, many people are there. Sonia, Trup.
Sir, I would like to ask one question.
Yes, sir. Please proceed.
Share your experience regarding whatever while you are preparing for your university examination. Okay. So simultaneously, how you focus for GPAT exam while preparing for your university examinations on same topics or subjects?
तर
Yes.
माझा पर्सनल एक्सपिरियन्स तर मी रोज अभ्यास करत नाही किंवा रोज बुक्स घेऊन बसणाऱ्यांपैकी तर नाहीच आहे डेफिनेटली. सो मी तर काय केलेलं, ओके, लेक्चर वेळी कन्सेप्ट क्लिअर करून घेतले होते, किंवा आपण नाहीतरी कोणीही म्हटलं तरी एक्झाम आल्याशिवाय नाहीतरी आपण थरली बसून अभ्यास करत नाही. सो जेवढे पण दिवस आपल्याला मिळतात, सेशन स्टार्ट और सेमिस्टर स्टार्ट झाल्यापासून एक्झामपर्यंत, त्या मधल्या डेजमध्ये कधी पण एखादं बुक काढा, आपले कन्सेप्ट क्लिअर करून घ्या. जो तुम्हाला चॅप्टर वाटतं, ओके आज हा चॅप्टर आपल्याला चालू आहे सध्या, ह्याच्यातला आपल्याला ओके हा टॉपिक नाही कळलाय, तर फक्त काढा, आपला कन्सेप्ट क्लिअर करा, तेवढं इनफ असतं फॉर प्री प्रिपरेशन्स ऑफ सेमिस्टर. कारण सेमिस्टरची जेव्हा PL लागते तेव्हा ॲक्च्युली आपल्याला खूप टेन्शन आलेलं असतं, आणि तेव्हा कन्सेप्ट क्लिअर करण्यापेक्षा ऑलरेडी आपले कन्सेप्ट क्लिअर असतील रेग्युलर स्टडीमध्ये तर ते खूप हेल्पफुल असतं. कारण मध्ये काय करायचं असतं, तर ऍक्च्युली आपल्याला युनिव्हर्सिटीप्रमाणे आन्सर ड्राफ्ट करायचं असतं. फक्त आपले कन्सेप्ट ऑलरेडी क्लिअर असले पाहिजे आपल्या रेग्युलर स्टडीमध्ये. सो आपले कन्सेप्ट क्लिअर करून घ्या रेग्युलरली. जीपॅटसाठी मी जसं सांगितलं तसं मी अगोदरपासून स्टडी करत नव्हती. मी लास्टच्या 1 महिन्यातच अभ्यास केला जो केला तो. त्यामुळे माझा असा काही एक्सपिरियन्स नाही आहे साईड बाय साईड मॅनेज करण्याचा. कारण जानेवारी और डिसेंबर तेव्हा आपले स्पोर्ट्स वगैरे सुरू होते, त्यामुळे तेव्हा माझ्यासाठी ते इझी झालं. बट सेमिस्टरसाठी प्रिपेअर करताना माझा हेच होता की रेग्युलरली जस्ट कन्सेप्ट क्लिअर करायचे, आणि PLमध्ये ओके बाबा हा पॉईंट इम्पॉर्टंट आहे, आन्सर ड्राफ्ट करताना हा पॉईंट आला पाहिजे आन्सरमध्ये, बस एवढंच करायचे
Okay.
मी.
What do you think about it, Prajakta?
सेम सर, मी पण कधी असा स्पेशली वेगळा अभ्यास केला नाही. आम्ही आधी एकत्रच अभ्यास केला. ग्रुप डिस्कशन मेथडनेच आम्ही जीपॅटचा पण अभ्यास करायचो, आणि सेमिस्टरसाठी सुद्धा मी हॉस्टेलमध्ये राहते. सो प्रत्येक सेमिस्टरला आम्ही ह्याच्या परीक्षेच्या आधी हार्डली 10 डेज आम्ही अभ्यास स्टार्ट करायचो. कन्सेप्ट क्लिअर असायच्या, क्लासरूम मध्येच होऊन जायचं सगळं, त्यामुळे स्पेशली करावा लागला नाही. आणि जीपॅटसाठी तर आम्ही जीपॅटचा स्टडी म्हणजे आता कसं झालं, नायपरचा स्टडी करताना आम्हाला 8व्या सेमिस्टरच्या सोबतच अभ्यास करायचा होता. त्याच्यामुळे 8व्या सेमिस्टरचे सुटिक्स किंवा मेडिसिनल केमिस्ट्री हे सगळं आम्ही रिलेट करत गेलो. हे सुद्धा आम्हाला सेमिस्टरला आहे आणि नायपरच्या प्रिपरेशन मध्ये सुद्धा आहे. सो अशा गोष्टी तुम्ही रिलेट करत गेलात तर ते दोन्ही प्रिपरेशन साठी इझी होऊन जाईल.
म्हणजे एक simple, we are always telling and you are giving a guidance, or always we are telling to our students, you have to do the comparative study, and that would be a more better for preparation such examinations like that GPAT and NIPER. One more question is there from Hina Kazrekar, final year student. Please discuss regarding the time management during the examination days. Miss, how we have or we can say they have to manage the time during examinations. Yes,
Okay.
time management.
टाइम मॅनेजमेंट नायपर वेळी तर आम्हाला नायपरला आता फुल लॉकडाऊनच मिळालं होतं ऑलमोस्ट 3-4 महिन्याचं. जीपॅट वेळी टाइम मॅनेजमेंट आय वुड से आम्ही काय केलेलं, सकाळी ठरवायचो की आदल्या दिवशी ठरवायचो की हा टॉपिक आम्हाला आज कव्हर करायचा आहे. एकतर सेम टॉपिक घ्यायचो किंवा टॉपिक डिस्ट्रीब्यूट करून घ्यायचो तिघांमध्ये, आणि तो टॉपिक आम्ही सकाळी वाचून यायचो. जेव्हा आम्ही सकाळी भेटायचो तेव्हा क्लासमध्ये असेल, रिसेसमध्ये असेल किंवा अजून काही, आम्हाला जेव्हा जमेल किंवा कॉलेज नंतर सुद्धा आम्ही 6-6 वाजेपर्यंत थांबायचो आणि ओके डिस्कस करायचो की हा पॉईंट आम्ही आज स्वतः स्टडी केलाय ना, मग एकमेकाबरोबर शेअर करायचो की ओके ह्यात हे इम्पॉर्टंट पॉईंट आहेत. देन डिस्कशन हे झालं सकाळच्या फेजमध्ये. त्यात नवीन अजून काय असेल ते सुद्धा बघायचो आणि रात्री जाताना ठरवायचो की ओके हा क्वेश्चन पेपर आज सॉल्व्ह करायचा आहे आणि आम्ही तो क्वेश्चन पेपर रात्री सॉल्व्ह करायचो. स्वतःचा स्कोअर डिस्कस करायचो व्हॉट्सअपवर आणि नेक्स्ट डे आल्यानंतर क्वेश्चन पेपरमध्ये बघायचो की बाबा हा बरोबर आलाय, सगळ्यांचा आलाय का, ओके नाही तुझा चुकला, मग तुला काय कन्फ्युजन होतं, मग ते असं स्टडी करायचो.
Okay, means this is a way of clearing the concepts. You are saying
Yeah.
this is the best practices that I can suggest, and I would suggest to our students also. This is the best way. Means suppose you are having any confusions, little bit difficulties to understand that topic. So better way you should share, and you should ask to your classmates or your, we can say, group mates, and that is the best practice. Okay. Anything else regarding this? Anybody want to ask?
तिने एक्झामच्या दिवशी नेमकं कसं टाइम मॅनेजमेंट करायचं असं विचारलं
I
होतं आय
think so.
थिंक.
Yes, yes, yes. You
सर
are right.
जीपॅट साठी तसं आपल्याकडे वेळ म्हणजे असतो, सफिशियंट असतो, त्यामुळे त्यात घाई करायची पण गरज नाही. जसं जसं आपण एक एक क्वेश्चन सॉल्व्ह करत जातो आय थिंक आपण आपला क्वेश्चन पेपर पुन्हा रिपीट करू शकतो, म्हणजे रिवाइज करू शकतो, एवढा वेळ आपल्याकडे असतोच. त्यामुळे
Huh.
जीपॅटच्या एक्झामच्या वेळेस टाइम मॅनेजमेंटची एवढी गरज नाहीये आणि
Okay, okay.
त्याच्यात
Because this is an online examination, na. You can see how many questions you have solved, how
हां.
many is pending, and how many you have answered like that. So that kind of thing is, I think, available. It is an online mode is there. So that is
हो.
that would be a more easier, okay, to manage your time during your examination. Yes.
कम्प्लीट
Anything?
पेपर सॉल्व्ह करून सुद्धा ना, माझ्याकडे तरी अर्धा तास होता कम्प्लीट माझा क्वेश्चन पेपर बघून परत चेक करण्यासाठी. काही जणांकडे 1 तास पण होता. त्यामुळे तुमच्या स्पीडवर आहे तुम्ही कसं सॉल्व्ह करता, कारण 125 क्वेश्चन्स इन 3 आवर्स नथिंग आहे. टाइम मॅनेजमेंटची गरज नायपरला लागते, कारण तिथे 2 तासात 200 क्वेश्चन सॉल्व्ह करायचे असतात आणि मला तरी टाइम पुरला नव्हता कारण काही क्वेश्चन्स लेंदी होते. सो टाइम मॅनेजमेंट हा प्रॉब्लेम नाही आहे जीपॅटमध्ये, बट नायपरला असू शकतो. सो स्पीड वाढवायची, क्वेश्चन पेपर सॉल्व्ह करायचे, दॅट इज ओनली थिंग यू कॅन डू टू इन्क्रीज युअर लाईक फिक्स की ओके बाबा 1 तासात मला एवढे क्वेश्चन झालेच पाहिजे किंवा 1.5 तासात माझे एवढे क्वेश्चन झालेच पाहिजे. असा टाइम लिमिट जर तुम्ही ठरवून घेतला तर इझिली टाइम मॅनेज होईल.
Okay. Yes. Anybody
म्हणजे
want to ask
म्हणजे
any
आता
questions?
प्रिपरेशन करताना तर ठरवलं की मला आजच्या दिवशी एवढे एवढे क्वेश्चन सॉल्व्ह करायचेच आहेत, म्हणजे मॅक्झिमम, म्हणजे असं अलॉट करू शकता की मला आज 50 की 100 क्वेश्चन्स करायचे, तर ते पण होऊ शकतं. म्हणजे एक्झाम येईपर्यंत तेवढे मॅक्झिमम क्वेश्चन सॉल्व्ह होऊ शकतात.
Yes. Okay. Yes. Anybody? Thank you. Yes. Anybody else? Anybody? Hello. Many of the students are there. Third year, final year students. Yes. Nagesh. Mr. Shubham Narkar from third year. Saili Satardekar from final year. Swati. Ankush Chavan. Yes. Anybody else? Okay, from my side, after all this discussion about the how to prepare for GPAT and NIPER, what you would like to suggest, Miss? Finally, Miss, what they have to do? So definitely they will focus towards the GPAT as well as the NIPER in one sentences or two sentences. What do you think, all of you?
प्रोसेस एन्जॉय करा, दॅट इज ओनली थिंग आय कॅन टेल. जस्ट अभ्यास करताना पण ओके मस्ती मजा करा. त्यामुळे तुम्हाला एखाद्याने जोक मारलेला पण क्वेश्चन ना, बेसिकली तेव्हा नेमका तो क्वेश्चन पडतो एक्झामला आणि आपल्याला आठवतं की ह्या माणसाने त्याच्यावर जोक मारलाय. सो एन्जॉय करा. एन्जॉय करत जेवढं कराल ना तेवढंच तुमच्यासाठी इझी पण होईल आणि तेवढीच तुम्हाला पण मजा येईल. त्यामुळे टेन्शन घेऊन अभ्यास करू नका, फ्रीली अभ्यास करा.
Yes, learn with fun.
या लर्न विथ फन.
Yeah.
येस.
Yes, Mona.
आणि कॉन्फिडन्स पण अजिबात लूज करू नका, वाचत राहा, ट्राय टू अंडरस्टँड आणि आणि काही गोष्टी ज्या लक्षात ठेवायच्या आहेत त्या लक्षात ठेवा. असं तुमचा अभ्यास
Consistency.
आपोआप होत जाईल.
Okay, Miss, consistency is required.
हो,
Okay, and
कन्सिस्टन्सी.
confidence. Yeah. Okay. Okay. Yes,
हो.
Prajakta, would you like to add anything else, Miss? In a one sentence, what you would like to suggest to your juniors for preparation of GPAT and NIPER? Yes, Prajakta, are you there?
येस.
Ha,
स्टडी
yes.
वेल बिकॉज ऍट द एंड तुम्हाला असा रिग्रेट नाही वाटला पाहिजे की आपल्या हातात वेळ होता आणि मी अभ्यास केला नाही, जरासा अभ्यास केला असता तर क्वालिफाय झाले असते. त्याच्यामुळे त्या मोमेंटला हा रिग्रेट वाटून काही फायदा नसतो. सो स्टडी वेल, स्टार्ट फ्रॉम टुडे ऑनवर्ड्स अँड ऑल द बेस्ट. सॉरी, बेस्ट ऑफ लक. लक इज रिअली इम्पॉर्टंट.
Okay. Yes, anything else? So thank you so much, dear alumni. Okay, Miss Pritam Palkar, Miss Prajakta Kashadikar, and Miss Mona Mohite, for your valuable, experience as well as the guidance for how to prepare for your GPAT as well as the NIPER examination. So I'm really thankful to all of you. I'm very much thankful. I would like to thank our honorable management, our principal, Honorable Dr. V. A. Jagtap sir, HODs Dr. Shinkar sir, Dr. Datta Prasad Shinkar sir. Then along with that, Mr. Rukari sir, and I would like to thank Mr. Mude sir for his cooperation. So thank you, and see you tomorrow, dear participants. Same, that is a series 2, and see you tomorrow. Thank you.
Thank you, sir.
Yes, I would like
Thank you,
to
sir.
tell you one thing. Just you can share your put your videos on for getting one snapshot. So we have to capture one snapshot. Yes, all of you, please put your video on, share your video. So that will be a okay. As a just yes, please, all of you. Those who are the participants. Okay. Thank you. See you tomorrow for same uh, series 2. Okay, alumni talk. See you tomorrow at 3 p.m. Okay, so the time schedule will be there from 3 to 5. Okay, so hopefully everybody will be there and enjoy. Okay, happy learning and keep learning.